AP 120, Chapter 9, Topics, Neuroglia and Neuron Basics. So, the structural division of the nervous system is based on two concepts: the central nervous system, which is the brain and spinal cord, protected by, of course, the skull and vertebral column, and then the peripheral nervous system, which is all of the nerves coming off of the spinal cord and brain, and reaching out, extending to the various parts of the body. And that includes cranial nerves coming off the brain and spinal nerves coming off of the spinal cord. The functional division is that we have a sensory division where sensory receptors detect change in the environment. That information gets sent along the sensory division to the central nervous system. There, that information is integrated, processed, and a response is determined. Then that response is sent via the motor division being broken down into two smaller parts: the somatic nervous system, which controls the skeletal muscles, and the autonomic nervous system, which controls smooth muscle tissue, cardiac muscle tissue, and glands. Here is a slide of nervous tissue. This large cell here is the neuron, and then all these little dark spots, they represent the nuclei of the various neuroglia cells. So here is a basic neuron where we have the cell body. Cell body houses the structures expected for cell function, nucleus, mitochondria, Golgi apparatus, etc. Then coming off of it are these short little processes called dendrites, and the dendrites and the cell body are able to receive signals, so they are receptive to receiving signals. And if a signal is received and response is needed, then this signal will go through this very, very long process called the axon, and will travel down to the ends of the axon, where it would then pass that signal on to the next cell, whether it's a neuron or an effector. So when two cells come together, that is the synapse. So when the neuron comes together, forms a synapse with either another neuron or an effector cell. And the signal, the information, gets transmitted through this tiny space between the two cells called the synaptic cleft. So the nerve impulse itself cannot cross that space. Instead, chemicals are released. These are called neurotransmitters. They are released at the end of the axon for one neuron, and then they diffuse across the synaptic cleft and then bind to receptors on the second cell. And this should look familiar. This arrangement is identical to the neuromuscular junction because that was a type of synapse. All right, moving on to the neuroglia cells. Turns out there are more non-neuron cells in nervous tissue than there are neurons, and they carry out a variety of functions in order to aid and protect the neurons. Some of their functions include supporting the neurons, physically supporting them, controlling what is in their environment. They provide structural frameworks for the nervous tissue. They can produce the myelin sheath, which wraps around parts of the neuron. They can be engaged in phagocytosis to, say, clean up cell debris or get rid of pathogens. And most importantly, neuroglia cells can divide. They are able to undergo cell division. In the central nervous system, that's the brain and spinal cord, there are four types of neuroglia cells. You have microglial cells, oligodendrocytes, ependymal cells, and astrocytes. The microglia cells are relatively small. They move around in the nervous tissue, and they engage in phagocytosis to engulf bacterial cells, cell debris, and so on to keep the tissues clean and safe from pathogens. The oligodendrocytes, on the other hand, look a little bit like, octopi. They have these processes that extend out to regions of neuron axons. And what they do is they wrap around that part of the axon to form the myelin sheath. And the myelin sheath is extremely important because it speeds up the nerve impulse, speeds up that action potential so it gets to the ends of the axons faster. Then we have epidermal cells. Epidermal cells line the ventricles, which are open spaces in the brain, and they form what are called choroid plexuses with blood capillaries, all with the purpose of producing cerebral spinal fluid. So epidermal cells produce cerebral spinal fluid. And then finally, we have the astrocytes. Astrocytes have a lot of different functions. They provide physical support to neurons. They make contact with the neurons. They help to hold the neurons in place, guide the neurons in growth. The astrocytes also regulate the nutrients and ions found in the fluids surrounding the neurons. If nervous tissue should take damage, the astrocytes will divide and fill that space up, forming a kind of scar tissue. And the astrocytes form the blood-brain barrier. So the astrocytes send out processes that then cover the blood capillaries, making physical contact with the blood capillaries and limiting what can leave the bloodstream and enter the nervous tissue. And this acts as a shield to block entry from a lot of different kinds of compounds, pathogens, and so on, to isolate and protect the neurons as much as possible anyway. In the peripheral nervous system, out among the nerves, we have the Schwann cells. And the Schwann cells do one thing, and that is produce myelin sheath. So a single Schwann cell will wrap around a portion of an axon, and then it'll wrap many, many times to form the myelin sheath. And as expected, the myelin sheath will speed up the nerve impulse. So it'll speed up the action potential, cause the signal to get to the ends of the axon much more rapidly. So here again is the parts of the neuron, the dendrites, the cell body, the axon. Receiving ends, the dendrites, sending out the signal, the axon. Dendrites again are short and usually branched. They are highly receptive surfaces, so they form synapses with other neurons. So they are the receiving end of a synapse. And they have many receptors for neurotransmitters, for those chemicals released at the ends of the axons. And when they are stimulated by the binding of the neurotransmitters, they will send a signal toward the cell body. Cell body, again, houses all the standard organelles that we would expect. It includes the nucleus, mitochondria, rough endoplasmic reticulum, and so on. It also houses neurofibrils. Neurofibrils are long, thick protein filaments that are a specialized form of cytoskeleton that travel from the cell body down the length of the axon. And neurofibrils are important for carrying materials made in the cell body to the ends of the axon. Also found within the cell body is what's called chromatophilic substance. So chromatophilic substance is basically a modified areas of endoplasmic reticulum, and they are grayish in color. So chromatophilic substance is grayish in color and involved in protein synthesis. And here is the very long axon. Axon, of course, has lots of neurofibrils running down its length. The beginning of the axon is called the axon hillock. That's where it's coming off of the cell body. Then it stretches various distances until it gets to the very end of the axon, where you have a little widened area called synaptic knobs. And it's the synaptic knobs at the end of the axon that forms the synapses. It's worth noting that axons can be extremely long. You could have an axon going from your spinal cord down to your toes, and that's like what, 3 feet long? Pretty crazy. And as always, the axons are carrying a signal away from the dendrites and cell body toward the synaptic knobs to then pass that information on to the next cell at the synapse. Again, myelin sheaths cover many regions of the axons. Myelin sheaths can be formed by Schwann cells in the peripheral nervous system or by oligodendrocytes in the central nervous system. They act as an electrical insulator, isolating it from electrolytes, and in the process speeding up the neural signal, speeding up the action potential as it travels down the axon. To form the myelin sheath, the cell, whether it's Schwann or oligodendrocyte, will wrap around that portion of the axon again and again and again, so you get a very thick layer of plasma membrane. Now, the myelin sheath does not cover the entire axon. There are gaps between portions of the myelin sheath. These gaps are important. They're necessary for the function of the axon, and they are called the nodes of Ranvier. So the gaps in the sheath are called nodes of Ranvier. Now, most neurons are myelinated. Over 90% are myelinated, but there are some out there that are unmyelinated, that do not have a myelin sheath. All right, here is another view of the myelin sheath. As you see here, the cell wraps many, many times to form a thick layer of plasma membrane, highly insulating. Got the nodes of Ranvier. And it's worth noting that myelination is white in color. So the myelination makes the axons whitish in color. Why does this matter? Well, when you look at, say, the spinal cord or the brain, you can see areas that are grayish in color, called gray matter, where you have lots of cell bodies, lots of synapsing occurring, and it's gray because of the chromatophilic substance found in the cell bodies. And where you have lots of synapsing is where the hard work, the processing is occurring. So you can think of gray matter as the hard drive of the central nervous system. And then white matter is we have lots and lots of myelinated axons. And myelinated axons are carrying signals from one place to another. So they are acting as connecting wires. So white matter has lots of myelinated axons because of the myelination being white in color. You can also see that the gray matter in the spinal cord is toward the inside, while the gray matter in the brain is toward the outside, toward the surface. All right, there are a few different kinds of neurons structurally. You have multipolar, which is exactly as we described, dendrites attaching to a cell body, then forming an axon. This is extremely common, all neurons in the central nervous system and many in the peripheral nervous system. Then we have bipolar. Bipolar, you have some dendrites forming a long structure going to the cell body, which then leads to the axon. Bipolar neurons are relatively rare, and they are found specifically in the nerve tissue of the eyes, the nose, and the ears for detecting those sensations. And then the third structural type of neuron is the unipolar neuron. Here the dendrites go straight to the axon that leads then to the end of the axon. The cell body is off to the side and not involved in the signal. So again, in unipolar, the dendrites go straight to the axon, and the axon does not need the cell body. It's off to the side. Unipolar neurons are the sensory neurons in the peripheral nervous system. So they are somewhat common. They are the sensory neurons in the peripheral nervous system. Ganglia. Ganglia are widened areas where you have lots of neural cell bodies in the peripheral nervous system. So for instance, the dorsal root ganglion is made up of neural cell bodies, specifically the unipolar ones. So that's where they keep the cell bodies for those unipolar sensory neurons. Ganglion, masses of neural cell bodies in peripheral nervous system. And that is it for this part of chapter 9.